Welcome to this video from the University of New Hampshire Cooperative Extension. In this video, we will cover the proper methods for collecting plant tissue on greenhouse crops to be analyzed for nutritional status. There are two reasons why we conduct nutritional testing on greenhouse crops. The first is to monitor the nutritional status. In this case, we routinely sample weekly or monthly depending on the crop. The second reason is to diagnose nutritional symptoms. We do this when we first start seeing symptoms of a nutritional disorder. When sampling, subdivide your greenhouse if there are differences between crops, irrigation zones, or symptoms. You can see in this photo that there is a striking difference between these two benches. Be sure to sample these separately. Also, sample symptomatic plants and then sample healthy plants separately to use as a comparison if you're trying to diagnose a problem. When sampling, it is important to sample a crop across the bench in a pattern shown here in red to ensure to eliminate any variability there may be across the bench. Be sure to check with the lab that you're using as to what leaves you should be harvesting. But in most cases, we want to harvest the most recently matured leaf on a plant, like the ones that you see here. Leaves to avoid are the older leaves at the bottom of the plant or the youngest leaves at the top of the plant. Here are examples pulled off of the plant. We want to harvest leaves like this one and avoid the others. Also avoid leaves that are profoundly damaged. One last thing to consider is the stage of symptoms. Here's an example of a leaf that is just starting to show symptoms of a nutritional deficiency. This is the best time to sample for a diagnostic purpose. To collect your sample, simply walk up and down the bench and harvest one leaf from an individual plant, and choose plants up and down the bench in the pattern discussed earlier. Take care when removing the leaves so that you remove the entire petiole, but do not damage the plant. Here's an example of how not to remove the leaf. In this case, some of the petiole is left on the plant. This remaining portion of the petiole is the prime opportunity for disease like botrytis to infect the plant. In most cases, about 1 ounce of fresh tissue is sufficient for a sample. In this example of poinsettias, that equaled about 30 leaves, or one large handful. The leaves may have dust or other residues that might interfere with the analysis, so we need to wash them. Simply bathe the leaves in deionized or distilled water and gently agitate for about 30 seconds. Remove the leaves from the water and shake off any excess water. Place on a paper towel and blot the leaves dry. While the leaves are drying, clearly label a paper bag with its appropriate information. Be sure to use a paper bag, not a plastic bag. Tissue placed in a plastic bag will most likely rot in transit and not be usable. Carefully place the leaves in a labeled paper bag. Most labs will provide you with a submission form. Be sure to fill this out completely, including your name and the crop and any other pertinent information. Finally, place your sample along with the submission form in a padded envelope or box and send it to the lab. It is best to use overnight or second-day delivery to ensure that your sample does not deteriorate over time. Thanks for watching this video on taking a plant tissue sample. For more information about growing in a greenhouse, visit the University of New Hampshire Cooperative Extension's website at www.nhfloriculture.com.